So welcome to our Minflux workshop. We'll get started. We have about almost 30 people registered, so about half of those are online. And so you'll see us kind of talk about the core maybe a little bit more than we usually do. Okay. So here's our schedule. I'm Glenn Freed. I'm the director of the core facility. I'm going to give a little bit of an introduction to Minflux and talk about the instrumentation. Kingsley's going to talk about sample prep, and then we have some people who have used our instrument in the last 3 months talk about their data. And so the point of this workshop is to let new people know about how the instrument works, what they can do with the instrument, and we're trying to build a situation where people from off campus can bring samples and run our Minflux. After the coffee break, Kingsley will run a live demo, and so some people can stand in the Minflux room for that and watch what he's doing, and we will have it also in this room on Zoom. So if that room is too full, we can do that. Okay, so a little bit about the core. For people who don't know about the core, we have about 300 researchers that come to use the core each year. So some of these people, most of them are graduate students, some are postdocs. We have about 10% undergrads. About half of them are engineers from the north side of campus. The other half are biology-based people. And they come from 125 different research groups. So this should tell you that there's a lot of different kinds of research going on in the core. For people on Zoom, I put this little map up. So we're kind of in the center of Illinois over here. It's a little ways to St. Louis, a little ways to Indianapolis, and a couple hours to Chicago. So we're kind of in the center there. We have a variety of instruments. We have 3 confocals that people can use now. We just purchased an LSM 900 about 2 weeks ago. So it will be here the end of January, or the end of December, beginning of January. We also have 2 slide scanners, an AFM serial block face SEM. We just had a nice lunch with the core. Well, actually the last 2 weeks of lunch with the core had serial block face SEM data. We have a X-ray CT system for big samples, and we have some other instruments there. So now we're going to talk about our Minflux. Oh, this is the core staff. Kingsley is the person in charge of the Minflux at the moment. Austin does a lot of instrumentation stuff. Dewey does training on some of the confocals. Miranda does training on the slide scanners. And Hannah takes care of registering people and answering questions and setting up conferences and workshops. So on to the Minflux. We purchased the Minflux with Martin and Zan, so they're 2 faculty on campus, and without them, the Minflux certainly would not be here today. There are other couple groups we like to thank is the campus units. And so on our campus, you may not realize this, but units really, in my mind, work together very well. So about half of the money to purchase the $2 million instrument came from campus units. And that happened by Zan and Martin going and talking to people, unit heads, and saying we'd really like some money. And that is very nice to be able to purchase things quickly when people are willing to put up money. And the rest came from the Roy J. Carver Charitable Trust. And so we thank those people also. So this was installed the end of June, and so that's a few months ago. And so you'll see how far we are in a few months. If you have questions, we have a website. Feel free to Google us. We have our emails and contact us. Okay, so now we're going to talk about Minflux. Obviously, the reason we're here is because people are interested in 2 nanometer resolution in fluorescence imaging. And so this is an extremely special resolution, a capability, right? So whenever I look at this, I think back, I've been doing this for a few years. Most confocals have 250 nanometer resolution. This is 100 times better than that. And we can locate particles within 100 microseconds. And that's very fast, right? So this is these are tracks. This is a molecule that the resolution has never been seen before. And so you're going to see this data like this in a little bit. So now I wanted to talk about super resolution, kind of give a little bit of background on super resolution imaging, fluorescent imaging. So I put the scale bar in here so we can think about what we're doing on scales. And what we do is we put some labels up, right? And an easy way to try to image this structure is to use something like confocal, and you scan a beam over your sample, and you can easily see that your resolution here is around 250 nanometers, and it's not going to separate these molecules, and you're not going to see this shape at all. The second thing you can do is do single molecule localization, and so this is a well-established technique. The first thing you do is you typically turn on a single fluorophore, and then you image that fluorophore, and so now you take a wide field image of the whole area. Somewhere on your camera, you see only photons from this fluorophore, and you can get about 10 to 20 nanometer resolution here. And you need about 20,000 photons to do this, so somewhere in this range. So these numbers vary a little bit, but the point here always is that you're scaling by 1 over the square root of the number of photons. So you're in a competition of burning out your fluorophores before you get the resolution you need. So we're here to talk about Minflux. So this is where we're going to talk about that. And so the way Minflux works is you first turn on a fluorophore, kind of like we did before, and we use 405 nanometer light. But now instead of flooding the whole area, you're looking at a smaller region. And then you excite that, but instead of exciting the whole region, you use a donut. And so I put this little mark here to mark the center of our donut. You get a certain number of counts, and the counts are related to how far you are from the center of your donut. So if you get out here further, your counts are going to be higher, and there's some noise here, right? So the advantage of Minflux is that you can move your donut around and you know where your donut's at. So now you can move over here, and now we get more counts. You can move up here, and you get a few more counts. You can move over here, you get another number of counts, and you move down here. And so you can see we're making a pattern, and everywhere in our pattern we have a different number of counts. So now at the end of that little process, we call this our first iteration to localize our fluorophore. You can come up with an idea and an estimate of where your fluorophore is. And now your ability to locate this is related to the size of your pattern and over 4 times the square root of the number of photons. So here there's a couple advantages. One is there's this little change down here, but the big advantage is that you have this L, which is related to this size, right? And so when we're running our Minflux at the moment, we do iterations that start with L around 300 nanometers, and they go down to 40 nanometers. So you do this once at 300 using the galvo scanner to move the donut around, and then we have electro-optic deflectors that move this around again at 300 nanometers, and then 170, and then 75, and then 40. And so you can see as this L shrinks, now your resolution is getting much better, much faster than if you just park your light on top of your fluorophore. So here we are at a smaller scale, and we get new numbers. And then this is kind of the light path that we have. So I wanted to go through this a little bit. So we have, so first off, so this is the Minflux light path. So first off, we have 2 excitation lasers that we can use. This is the electro-optic deflectors here. So now we can move our beam up and down quickly. So the choice of these is tied into that 100 microseconds to move your beam around on your sample. So then the light comes down here. We have a spatial light modulator here that we use to make the donut. Light comes in here, and we have a deformable mirror here. And so when I was describing this pattern, it was a 2D pattern, right? And we can also do a 3D pattern by changing this mirror here. And on the 3D pattern, instead of using 6 points around the side, we use 4 points, and then we use 1 on the top and 1 on the bottom. So you still have a center plus the 6 outside points. And so here's the galvo scanner, here's the microscope. So that's the excitation path, goes through like that. Light goes up to your sample, you collect photons like you expect, things come back. They come back through here. The 405 nanometer laser is the one to activate your fluorophores. So before you do your search pattern, you turn this on, and then when your emission light comes back through a pinhole, so that blocks out a focus light. So this doesn't have to be at the same level as a confocal, but it blocks room light and background signal. And then we have 2 APDs that we use when we're doing Minflux, and they're split by a dichroic. So the reason you have 2 APDs is because you want to do 2-color imaging in Minflux. In a confocal, you always do 2-color imaging by using 2 different excitation lasers and then a dichroic, right? So here, it's hard to have two donuts. When you locate a molecule, you could locate the molecule with the wrong laser, for example. Oh, that's one problem. And a second problem is it's very difficult to get your 2 donuts to overlay each other. So the way around that is to use 1 excitation laser, put your dichroic here, and then as you're collecting photons, keep track of the number of photons that come from here and here. And so when you do your localization, you're adding these 2 together. When you decide which color your image or your fluorophore comes from, you take a ratio from these. And so here's the typical example. You can see slight shifts in the two fluorophores you normally use for imaging, and then you find this ratio, and that's how you separate two colors in Minflux. So here's a few things that are important to notice about the usability of our system and the system that Abberior sold us. So first of all, it's obviously set up with an Olympus wide field microscope. It's an IX81, so all of the normal, when you watch Kingsley use the system later today, it's like using a normal microscope that you're used to. It also has a complete confocal setup. So many people are used to walking into a core, putting a sample on, finding the location they're interested in, using confocal, right? And so that you can do that same thing. You can actually use all these lines for confocal. 405 is a little bit of a, we suggest not using a 405 label for confocal imaging, because then when you're doing Minflux, you get out of focus light. But the rest of these work fine for Minflux samples. In Minflux, we can use the 560 nanometer laser or the 640. The 640 works best for the two-color imaging. The 560 you can use for tracking, or you can use the 640 for both of those. And then the other important thing that makes this different than other microscopes is this stabilization. So typically, if you put a microscope on an optics bench, you're going to have drift on the order of a quarter micron or so. You can see drift in a confocal over time, for example. To measure, to locate molecules to 2 nanometers, you need to have drift that's significantly lower than that. So these numbers here came from the Stefan Hell paper, and it's orders of magnitude lower drift than in a normal IX81. Okay, and so this is the light path for the drift compensation. So this is the way that the drift compensation works here. So many people, like if you have a Zeiss, we have a lot of Zeiss microscopes, and we have a focus drift correction on those. And the way it works is you reflect a light off the cover slip, and you look at the intensity of that, and you keep it the same. That works to some degree, but it doesn't work to sub-nanometer levels. There's no way that's an order of magnitude off. So what they do is slightly more complicated. They start with 980 nanometer light, comes through here, and they do some polarization control. So the polarization's out of the page. They then have a beam splitter, so the light is reflected in here, goes through a quarter wave plate, and then the light comes up and reflects off gold beads. And then the image comes back down. We're rotating in a different direction now. We go through a 4F relay system, and this spatial filter is crucial, so it's hard to see, but it's this little block. And the spatial filter blocks out the low frequency information. So their gold beads end up looking like this. So this, normally without the spatial filter, gold beads would look like a round spot. Here they've removed the low frequency information in this Fourier plane, and what they're left with is the high frequency information. So now they can tell, as you focus up and down through the beads, this shape turns from vertical to horizontal. So by looking at this image, you can tell where you're at. The other thing the spatial filter does is in removing all the low frequency noise or low frequency information, all the out of focus beads here are mostly made up of low frequency information in the image. So now you're tracking beads that are very close to the focal plane of your microscope. So that's important because if your sample's shrinking or expanding on the nanometer scale over time, you're ignoring the information you don't want to have and you're focusing on the information you do want to have. So they take, and when you lock your stabilization, they take a series of images and they use this piezo stage up here to take images on nanometer scales going up and down. So they have a library of images of where the gold beads are at each location. So now they can correct in X and Y and Z based on these images. And so they're continually having a feedback control by looking at the image that they're collected, compare it to a library image, and then move the stage accordingly. Okay, so now we're going to look at the actual physical instrument. This instrument's always been kind of interesting to me because most of the time when you buy a confocal or an optical microscope, somebody came up with the idea and then they did a lot of work to change it into manufacturing. This came looking just like what they built on the lab bench in Germany. So that's kind of interesting to me. Here's the Minflux lasers, the 640 and 560. The beam paths are combined here, so they use the same electro-optical deflectors here. You go through one in X, one in Y, come out through there. And then, so that's this section here. You come down here, and your beam travels together, and then it's separated with a dichroic, and the 560 line goes to a spatial light modulator set up for that wavelength, and there's a 640 spatial modulator here. So the reason you do these is because you need to change the phase in a pupil plane to create the donuts, right? So if you change the phase so it has a pattern like this on this spatial light modulator in the focal plane on the microscope, you get a donut that looks like this in X and Y and this in Z. If you do a top hat function at the same spatial light modulator, you get what we use for 3D localization. So it has high intensities here and then low intensities in a donut shape around the center that looks like this. Here's the deformable mirror, and so that is this part over here. So both colors come together through the deformable mirror. The point of the deformable mirror is to quickly move the focus up and down. So when you're trying to do 3D localization, you need to do a point above and below your guessed location, and you need to move up and down. So this is faster than running the piezo stage up and down. It's faster than running the focus on the microscope up and down. So this is what gets you the high-speed 3D localization right here. And so later in the talks, currently, unfortunately, our deformable mirror is not working, so Kingsley won't be running 3D localizations today, but we have a mirror set up there, so we'll do 2D. The detectors look like this, so this is over here. In this case, you see there's only two. These other detectors are set up for confocal imaging, and then there's the two extras that we use here. And so it's worth thinking a little bit about the data you can get out of this. So first of all, there's a couple points I'm going to cover here. One is that it's possible to edit your search parameters. So currently we're using a search program that Abberior gave to us. We've asked them to change things, and they can do that for people, and it doesn't look like it's impossible to change. So that's something that's most of the time companies lock that down, and in this case it's not. The other thing you can do is collect a lot of tag information from each iteration. So when you do the 4 iterations to locate a molecule, you come up with all of this different information for each location. The advantage of that is when you look at your images, well, there's a couple advantages. One is that microscopists kind of feel a little bit uncertain about new techniques and new samples, right? And so you wonder if you're really seeing what's there. And so by going through all of this data, you feel better about it. You can see where the localization was for each step and see if it's getting closer or it's moving randomly around. You can find the ratios between the 2 detectors. You can see if the ratios between the 2 detectors is staying the same for each time it's localized or if it's changing. Yeah, there's just a huge amount of data, and we will have people using our instrument talk about some of this. And so, so this is the core staff picture again. Are there questions about how the instrument's set up when we got it and things like that? So we'll move on to Dr. Kingsley. He'll speak about sample prep. Oh.
All right. Once again, thank you for coming. And my name is Kingsley Boateng. I'll be talking about some applications of Minflux and sample preparation. And as Glenn indicated later on, I will do a live demo. And so we'll encourage you to stay around and see that it's very simple to acquire data. And our ultimate goal is to help people that are even not on campus to be able to send us samples, and you can actually control the instrument remotely. So we want to demonstrate all that. So you don't have to be at U of I to actually get your data on image, and then we can send back the data for you to analyze. All right, so Glenn has taken a lot of time to go through the principle behind Minflux, and so we now know that we can get 1 nanometer resolution. We can get 1 nanometer resolution, much better than other techniques like Palm and Storm, where we could get only 10 nanometer resolution. So we have actually based on what the company reports, we are now able to image molecules at a molecular scale. The other important point is that if we are able to now image at the molecular scale, then the main challenge now is labeling, because in microscopy, all that you're capturing is your label. Okay, it is It is about the labeling. So whatever you label, that is what you capture. And so if your labeling is not efficient, if you are not labeling all the details of the structure, then you don't get a much clearer view of what your tool is. So going back, if let's say we are interested in imaging this mitochondria, and somehow our labels are only scanty and highlighting just a few regions, then in the image that we get, this is what we get. We don't see the entire structure of mitochondria. So once you have a well-labeled structure, you have a better chance of perfectly resolving and getting a much more accurate image. All right. So the new limit is more about labeling when it comes to light microscopy because we've broken the diffraction limit barrier. All right, so based on Minflux imaging, we are now able to get resolutions of between 1 and 5 nanometers. Depends on whether you're doing 2D or 3D, and most importantly, it depends on your sample. Samples that have very high background may not be able to get you to the best resolution. Other issues that comes up is the type of labeling that you're doing. If you're using antibody labeling as opposed to nanotags or other smaller tags, you may be using a very big probe to try and image something very small, and so that could be a problem. So that can all affect your resolution. In terms of tracking, we are able to track events within 10 to 100 microseconds, as Glenn mentioned earlier on. And so this allows us to explore several areas that we couldn't explore earlier on. So we can get a much better 3D molecular maps of cells. We can get a much better idea of the structure of cell membranes, the nucleus and the nuclear architecture, nuclear pores, messenger RNA, mitochondria, endoplasmic reticulum. We can image Golgi vesicles, look at actin and microtubule movement, and also look at synapse. And for molecular dynamics, you can actually follow protein folding, interactions, RNA movement, and on some of those. So the rest of my talk, I'm going to look at some applications in imaging and then some applications in tracking. But just a quick recap of the imaging scheme. So Glenn explained in detail that if you're doing a single color, you just use one detector, and you must use a label that can be excited by a 640 laser. So for imaging, the only label that you can use are labels that must be excited by a 640 laser. So even though we have a 561, you cannot use that for imaging. That is for tracking, and we can discuss more about that. Now, if you are interested in two color, then both labels must also be excited by the 640 laser. So in this case, we have 647 and CF680. Both of them are excited by the 640 laser. But then this is the 647 emission and CF680 emission. So we put a dichroic here around 685. And then we have two detectors that are collecting signal for each. And so we use a ratio of one detector, the signal from one detector divided by the total. And so if the ratio, for instance, is less than 0.6, then we can assign that signal to 647. And if the ratio is greater than 0.6, I should say 0.6 or 0.4, we assign it to 680. So that gives you an idea of how two-color imaging is done. Now, as I indicated, assuming this is your structure of interest that you're trying to label, then you want to put dyes that are able to accurately label all the regions of interest, and to the best extent, you don't want to overlabel, but each of the areas must be well decorated or labeled. And so the dyes must be such that they can be switched on. One molecule can be switched on at a time. So you don't want to have a situation where all the dyes turn on, because this is single molecule localization. So our goal is to localize one molecule at a time. And so we go through, localize this molecule through, by going through all the 4 sets of iteration. We come back and we go on and localize the next molecule. So we do that until all the molecules have been localized, and then we reconstruct the localization of these molecules to get us the image. So keep in mind, we are only imaging one dye molecule at a time. All right, so the main issue now is how do you label your samples? So there are different techniques that are being used. One of it is based on the same approach that is used in palm. And so you can use photo-switchable organic dyes. And so these dyes, an example, the Genelia 546 or 646 dyes, which are sold by Tokris, those often do not have any fluorescence, but it can be switched on with the 405 laser, and so then a molecule is switched on at a time, and then you can image it. Then you can also use photoconvertible dyes, and so an example is maple is a very common one, and so these dyes can be excited, and then you can switch it from, let's say, a 488 to from green to red, and then you do your tracking or imaging based on the red activation that you are now detecting. The most common ones, and which seems to work much more easier, especially if you're not interested in live cell imaging, is using carbocynon-based dyes. So we have the Cy5, the Arto dyes, Alexa Fluor dyes, and Abberior now also have their own set of dyes. They call them flux dyes. So these dyes, and I'll go through the mechanism, oftentimes this is the fluorescent state, and they can be switched on to a dark state by binding a tile molecule to it. And so these are the type of dyes that seems to work very well. And so we recommend using one of these carbocynon-based dyes if you're interested in doing imaging. For these, most often it involves primary antibody and secondary antibody, and you get a huge probe. And so that is something of a concern. So a much smaller labeling is using a snap or clip, and this is the cell construct that you can make, and then you use a ligand to detect it. So a ligand that is labeled with a 647 to detect it. So you can do this oftentimes by fixing your cell and then adding the ligand to detect it. There are a few scenarios where some labels can penetrate into the cell, and you may be able to do some live cell imaging. But like I said, if you are interested and you are concerned about the resolution you're getting, you want to consider nanobodies instead of regular antibodies. And I bring that up because an IgG complex is about 50 nanometers in size. The dyes, the 647 or the organic dyes are very tiny, and so that is not necessarily the problem, but your antibodies are very huge. And so if you are interested in imaging things at 1 nanometer, 2 nanometer resolution, and a whole IgG complex can be about 25 nanometer. And so your molecule may be here, and then your probe is somewhere here. And so you don't really get a very clear representation of what the data is. Not that it doesn't work, but it does not always give you the best resolution possible. And so what we recommend is considering using snaptags or nanobodies. Those are usually about 3 nanometers. And so that you have a better chance of getting the best resolution possible. All right, so to make your sample, you want to seed your cells on an 18 millimeter cover slip. We recommend 1.5 or 1.5H. That is actually the best. You can use cover slips, or you can also use chambered slides. Now, you don't want to use any plastic, so this must all be glass. And then you do your fixation just like you do for any confocal or any other preparation, fixing with formaldehyde or methanol. And then, once again, make sure that the dyes that you select are excitable by the 640. And so the best dyes that we've seen so far are the 647, Alexa 647, or the Flux 647 if you're interested in one color. If you are interested in 2 color, then you may do any combination of these. So you may use Sci5 and CF680, or Flux 640 and 680, or Alexa Flow 647 and Flux 680. You can also use 647 and CF 680. That also works fine. But in your sample preparation, you want to keep an eye on your background. Backgrounds can be a huge problem because you are localizing single molecules, and any single signal in the background can affect your resolution. And so your goal is to make sure that you are minimizing the background as much as possible. And you can do this by increasing your blocking steps. So if typically you block 15 minutes, you want to do at least 30 minutes. And instances that I've done even 1 hour blocking. So you want to also increase the concentration of your blocking buffer if possible. So that is one way. You want to wash much longer and many more steps. So typically, if you're doing 3 washes for 15 minutes, do that for about 30 minutes. And then you want to decrease the concentration of your antibody. So typically, if for a confocal you do 1 in 100 dilution, you want to do 1 in 500 to start with, and I will probably even go to 1 in 1,000 dilution. And so labeling density is very important, and you want to really avoid over-labeling because that can create a lot of background noise for you. And once again, make sure you don't have any DAPI in your labeling, DAPI or whites, because if you are just interested in looking at the confocal, that is okay. But if you want to do Minflux, then obviously you don't want to put DAPI in your sample. So here is an image that I put up there, that before you come to do Minflux, you can look at your sample in a normal confocal, make sure that the signal to noise ratio is very high, so there's almost no background, and you can clearly get a signal from your labeling with no background at all. All right, so the principle that is used here is similar to the principle that is used in this term. In other words, we want to switch on 1 molecule at a time. And so the organic dyes that we use are these carboxanide-based dyes, which allows us to switch them on and off using a buffer. And so the buffer that we prepare, we call it a Glux buffer. It contains several components, but the most important component when it comes to blinking is the tile, and in this case, we're using MIA. And so what MIA does is that when you excite your molecules, the tile can then bind the fluorescent molecules and switch them off. So when you start imaging, typically you can use your 640 laser, increase it right away to maybe 10% to kind of bleach out or send the molecules into the dark state. You do that until you see only about 1 or 2 single molecules before you actually start your imaging. And so you send your molecules to the dark state, And once the molecules are in the dark state, then you want to find a way to turn them back on. And so you use your UV laser to then activate them again. And so this process is reversible. It can happen randomly, but then you can also use your UV laser to turn them back on to go from the dark state to the fluorescent state. And so by this, at any point in time, you are only looking at a few molecules, and you go through that whole iteration, and after a couple of minutes to hours, you can get a clear localization of all the molecules. But one important thing is the presence of oxygen. If you have oxygen in your buffer when you're mounting your sample, and you have a lot of oxygen in there, that can bleach your sample. So oxygen can move it from the dark state to the ground state, basically bleaching your sample. And so we have an oxygen scavenger system in the buffer that basically helps minimize that. And so I'll go over that in a few minutes. And so your goal is to make sure that you remove the oxygen as much as possible, and by that you can better control the kinetics of your blinking. And so that is the strategy that is used in Minflux. This is not unique to Minflux. As I said, D-Storm uses the same technique. All right, so this is the component of the glycolase buffer. And so if you're interested, we can send that to you. In the core facility, we have some that you don't have to make your own. But if you are a very heavy user, then of course you can purchase your own and prepare them before you even come. And so the components are glucose dissolved in trace buffer, and then catalase, you have glucose oxidase, and then the last component is the MIA. And so why do we have all this? Now we use the glucose and the glucose oxidase to eat up the oxygen. So glucose oxidase catabolizes the conversion of glucose to gluconic acid. And so that process, however, produces hydrogen peroxide, and you don't want to build hydrogen peroxide in your system. So we have catalase in there that also convert the catalase to water and then release 1 mole of oxygen. So here we take up 2 moles of oxygen, and then we release 1 mole. So at each round, you reduce your oxygen level by 1 mole. And so we have a huge concentration of glucose, and this process is cyclic. And so in the end, your net reaction is that you converting your glucose to gluconic acid and eating up your oxygen. And so the other important thing is that when you're preparing these samples, your buffer, you don't want to add them up until you are ready to image. And so you store the components in 4 degrees or -20, and then you only mix them up just when you're about to image. Storing these is very important, and then towing, you don't want to avoid free towing. So basically, whatever you'll be using within a month, you can keep that in 4 degrees, and then you can freeze, store the other components in a -20. All right, so basically, before you fix your cells, you level it, but the next thing is that you must add fiducials for stabilization, because we use gold bits to do stabilization. So we add about 100 microliters of gold bits to our sample. We incubate it for about 10 minutes, and then we wash it off. And so we wash it about 3 times with PBS. And then whilst doing this, we also prepare our buffer. And so we use cavity slides. And so it has a hole that can take about 100 to 150 microliters of the buffer. You add your buffer, and then after washing, you invert. So this is the incubation period. incubation period. After that, you wash your gold bit off, and then you prepare your buffer. You add your buffer about 100 microliter to the cavity slides, and then you invert it back onto, you invert your cover slip back onto the buffer, and then you seal it with this 2 mixture glue, we call it Twinsel. And so it takes about 10 minutes, and it's ready for you to image. All right, so for the next 5-10 minutes, I want to go through some examples or some applications. Some of these are published already. Some of them are work that we've done in the core facility. So this is one of the standard illustrations that Abberior uses to demonstrate the capabilities of the Minflux. And so these are nuclear pore complexes that are imaged in 2D. And so the nuclear pore complex is a cluster of 8 complexes. And so you see different clusters of these, and it is a 2-membrane complex. So you have the cytoplasmic membrane and the nucleoplasmic membrane. In 2D, obviously, you see only one. But the point of this image is to show that by imaging this, they can clearly get a 1 nanometer resolution. This is a SnapTag-based labeling. And so that is an endogenous system that is then fixed, and then you add the ligand, the 647 ligand, and then you can image it. This is now showing 3D of the same system, so 3D imaging. And so you see one layer and then another layer. And so this is actually a cartoon depicting the cytoplasmic membrane and then the nuclear membrane complex. And so here again in 3D, they can get about 2 to 3 nanometer resolution. And here are some claritin vesicles. And so this is done in 3D also, showing a labeling of PMP70 with ELISA floor 647. And so this is indirect fluorescence using regular antibody labeling. And here are some images of some neurons. And here again, this is spectrin, looking at it in 3D. So you see these nice ring structures in 3D. And one other thing that I want to mention is the 2 color. So here is an illustration of, so all the images that I've shown so far are 1 color. And this shows a 2 color. So this is a confocal image and a Minflux resolve image. And so we have the 2 colors, the green showing us the nuclear pore complexes, and then the red showing us DNA. And so one thing that I need to warn you is that you can see a very sharp contrast between the crispiness of the 2D imaging and then the 3D imaging. So for the same structure, you clearly see that the 3D is a little bit more challenging. Not that it's not doable, but it's a little bit more challenging. So keep in mind when you are interested in doing 3D imaging. And so here is another example of mitochondria. Here again is mitochondria TOM20, labeled with Cy5, and then mitochondrial DNA labeled with CF680. And so clearly showing that we can do 3D. Now, these are images taken from our facility. This is work done by myself with Peza and Dr. Kill. And so this project is interested in looking at the architecture of the meiotic chromosome. So here we have a confocal image. This is all the localizations, and then using the 2 detectors to separate. And so you see the green, that marks the outer cores of the meiotic chromosome, and then you have the red, that is a label that marks the inner core of the chromosome. And so Peza will be speaking a lot in detail about this later on. So this image is a 2D, And here is a 3D image of the same kind of architecture, now labeled with a protein. Once again, we have the outer protein that is shown in red, and then the green is showing a protein that marks the site at which the DNA are recombined. So typically, during meiotic division, the maternal and paternal chromosomes come together and exchange genetic material, and so we can see the sites at which the exchange is taking place much clearly. So now I want to talk a little bit about tracking. So tracking, like I said, you can use a 640, 560 or 640 laser. You localize your a single molecule and then you just follow it around. And so here are some examples. Once again, data provided by Abberior. And so here we are monitoring the lipid membrane that are tagged. And so you can clearly track that. And this is just image over 3 seconds. And then you can get your data, a lot of information that you can analyze. So typically you export your data into MATLAB or other programs like Python, and there's tons of information for you to do your analysis. Here is another example looking at bacteria, so tracking RNAs in bacteria. And so we have groups on campus that are doing similar work. And so this is a clear demonstration that that is doable. And then later on, Devinda will be telling us more about this. This is tracking microtubule movement. And so Kinesin working on microtubules. And so here again, this is image work that was done just before we purchased the system. They went to NIH and they did that, and then Devinda has done some work on campus, and so he's going to tell us more about that also. And some more tracking. This is from Martin's lab, and here again, Yuhan and Gopika will be telling us more about it. So this is tracking free RNA in cells, and so they are interested in the binding kinetics between RNA and proteins, and so they will be telling us more about this also. So as we indicated earlier on, our goal is to encourage people even outside campus. If you are in Chicago, you are in St. Louis, you are in Indiana, you can send us samples and we can image them. Actually, we can allow you to control the imaging yourself remotely. And so to ship us samples, we recommend that you put it in either a dish or a tube. You fill it with PBS and you seal it. And so you don't want to get the samples to us when they are dry. So as soon as you finish labeling, you put your tissue in a container, fill it with PBS, seal it, and then you can ship it in a normal cardboard. A room temperature shipping is fine. It doesn't have to be on ice. Typically, overnight shipping is fine. And so you can send it a sample, and then we right away mount it, and then we can do a Zoom or TeamViewer connection with you, and then we can select a region and then image. And so the software that we use is the inspector, and this is an overview of the software. We'll be going through this more during the live demo, but you can do some measurements in this software. Another software that we use often is also the ParaView. This is a free-based software that you can actually download on your computer. And this is very important, especially when you're interested in 3D rendition of your 3D data. And so this is the software that you can use to render your 3D data and follow the tracks of your tracking. So we'll be going through that later. So with this, I want to thank Abberior for providing some of the images, the core staff for their support in helping me manage the instrument, and thank you for being an attentive listener. Here again, tons of instruments that we have in the core, and so if you need our advice on any design of your experiment, you can come and talk to us. Thank you. Yes.
What's the total area that you can image?
Okay, so in a confocal mode, you can image 80 by 80. In Minflux, it's 10 by 15 microns.
And can you take your 80 by 80 and then overlay your Minflux right onto that?
Yes, so initially you have a confocal overview, and then the region that you're interested in, you clone that region. So you select your 15 by 10, clone that region, you have a confocal image of that region, and then you have another clone where you do your Minflux on, so that when you finish, you can have a confocal of that exact region as well as your Minflux image.
And then how much can you kind of, not the same, but could you file out from there?
Yes, you can do multiple regions at the same time.
Yep.
Yes, within a settings, yes.
And then has any of this ever been done on like super thin tissue slices before?
So we have not yet done this on like tissue sections yet, but I don't see why it cannot be done.
It might just be background, could always be.
It can be a background issue, but there can be ways to work around it, yes.
Okay.
Yes.
The same thing. So using multiple dyes, will it affect the background? Like let's say you have a 480, 560.
So 488 will not affect the 640 citation because it's far away enough. So if you want to look at your sample and with a 488 label, it's fine. You can add that. Even 561 is fine. But yeah, you can definitely add that. At least 488, yes.
And is there an optimal endogenous fluorescent protein combination for doing the 647, 680?
Yes, so in that case, you can look at SNAP and halo tags. Okay, so SNAP and halo tags, so you can do that combination and then detect each of them separately, yes.
Would that be the way, not actually using an FP like, like 12 or something like that, something that's more of a 680?
Yes.
So has that been done at all with endogenous fluorescent proteins against the halo and SNAP tags?
Yes, so the way the SNAP tag works is just like a GFP, but except that they don't have a fluorescent label. And so after you have your cells, you can fix it, and then you add a ligand. And so here again, you can have a 647 and a 680 ligand for the two different tags, and then you can image them.
And you can use those for live cell imaging as well, correct?
Some of them can penetrate, not all. So I believe.
For live cell imaging for looking at actual fluorescent proteins.
Yes.
Yeah, so that hasn't really been done that much yet.
Yes, so there are some surface markers that works fine, and I think there is one dye that Tokris claims can penetrate. We haven't tested it yet, but there is one that they claim can penetrate into the cell. But I can send you that information. Yeah.
That's kind of,
Oh, a question here?
yeah, that's kind of what you were just telling me.
Can you share a bit information on sample preparation for live cells? Yeah, I think that is what we just discussed. So if you want to do live cell imaging, you're doing a photo-convertible dye, a photo-switchable dye, or you can do the SNAP tag approach. And there are a few markers that can penetrate the cell, but oftentimes you may have to, if you have that limitation, that you may have to fix your cells before you can image them. One more question online. Okay.
Any Minflux image samples for actin?
For actin, yes. There's a guy in Paul Servan's group that has done some actin imaging. Unfortunately, he's not giving a talk today, but we've done actin imaging on actin and microtubules. Yes. Thank you.
Okay, so next we're going to hear about Kinesin tracking from Devinda.
is it? Okay. Is this your slides?
Should be. I actually might have closed it. It's in the desktop.
You can open it.
Thank you, Kingsley, for that talk, and thank you, Glenn, for the introduction. Hi, I'm Devinda Wijewardena, and I work in the Selvin group in the physics department here at UIUC, and I'll be talking about kinesin motility using Minflux. The key objective of this project is to observe kinesin under force and whether it undergoes limping, and for now we're looking at the early stages of this project with Minflux. So let's take an overview of the problem itself. So there's 3 different kinds of molecular motors we primarily focus on, cytoplasmic molecular motors in particular. And my group primarily works on kinesin and dynein, and this project will primarily involve kinesin. So kinesins transport cargo in the cytoplasm in one direction, while dyneins transport it in the opposite direction. And so the general experimental approach is to a lot of these molecular motor experiments is you have the plasmid for the motor. You, using site-directed mutagenesis, add mutations at a given site. Usually it's a solvent-exposed site on the motor itself, and then express the protein, after which you label it with an organic fluorophore. Usually you label it with either ATO647N, LD655, but there are other fluorophores you could label it with. You could even label it with a quantum dot, either on the foot or on the C-terminus. And so let's take a quick look at some of the existing problems in the literature in this area and why that motivated us to do kinesin tracking and where Minflux comes into the picture in that context. So one of the earliest controversies in this area was on the nature in which kinesin stepped when it had cargo on it. So the biggest controversy was whether it walked in a hand-over-hand fashion, so whether it walks like a normal human would, or whether it walked in an inchworm fashion, so as the name suggests, as an inchworm would. A former grad student in our lab, Ahmed Yildiz, who's now a faculty member at UC Berkeley, did an experiment where he labeled the foot of the kinesin and observed it walk under a microscope using a method that was developed in our lab called Fiona. So that stands for fluorescence imaging using 1 nanometer accuracy, Fiona. What he was able to observe was that kinesin took 16 nanometer steps, which is consistent with the hand-over-hand model, because had it been an inchworm model, he would have observed 8 nanometer steps. So this was the first question that was resolved in the area, but there were additional questions. For example, what were the intermediate states that occurred during stepping? So in between it taking steps, how did the stepping take place, and how did it correlate with the presence of ATP or ADP on the kinesin itself? And a lot of the early studies in this area that yielded breakthroughs involved the use of optical traps. For example, a study in 2017 from Eric Schaefer's group in Tübingen, Germany, they used an optical trap with germanium-coated nanospheres, and they proposed the presence of an intermediate step, so an 8 nanometer step while the 16 nanometer step happened. But for us to observe this in a super resolution microscope, we need better time resolution, so in the scale of 1 to 2 milliseconds, because ATP hydrolysis and these intermediate steps usually happen in the time scales of 8 to 10 milliseconds. We also need better spatial resolutions. So 1 to 2 nanometers, because if we are to observe an 8 nanometer intermediate step, we need spatial resolutions on the order of at least 2 nanometers. If we were to achieve these specifications with a Fiona setup, as Glenn had in his original introduction, we would have needed, like, to get 1 to 2 nanometer spatial resolutions with a 1 second exposure time, we would need about 10 to the power 4 photons. So scale it down to 1 to 2 milliseconds, you're going to need 1 million photons, which would mean almost instantaneous photo bleaching. So Minflux, on the other hand, helps resolve these issues because you're able to achieve these same spatial and temporal resolutions simultaneously with minimal photon fluxes, as the method's abbreviation suggests. So let's take a look at the typical workflow that I use for my kinesin tracking experiments. So first you have the confocal image. I have a customized data acquisition template. So this right here is the LD655 channel, so the red channel. I use LD655 as recently I started switching to ATO647N, but for these experiments I used LD655. So here you have the red channel, and then you have the microtubules here, which are labeled with TMR, which is excited by the 560 laser. And I've also got fluorospheres that are excited with the 488 nanometer laser. And I use these as fiduciary markers of sorts so that I could see the imaging plane clearly. Because sometimes with microtubules, especially if there are residual free-floating microtubules, it could be very confusing when you're trying to locate the imaging plane. So the 488 nanometer excitation beads help us locate that plane because they bind to the surface of the imaging chamber itself. So you have the kinesin that's labeled with the fluorophores, and in the presence of ATP, they walk. So once you have a region of interest selected, you can set that as the Minflux tracking zone, and then the Minflux tracking would occur. Over time, you'd have kinesin tracks form on microtubules. So as you can see here, this slightly slanted microtubule, and that's visible here. And there's also additional microtubules, and you see these tracks form. These are all kinesins walking. And the Minflux tracking algorithm, one by one, locates each of these, and it generates all of the tracking information. Now, the Minflux software, so Inspector, generates these Minflux files in a native MSR format, but we can generate additional data sets, so additional tracking information using either the .mat, so the .mat format or the .npy format. The mat format allows you to analyze the data in MATLAB, while .npy would allow you to analyze the data with Python. In our lab, we have a hybrid approach where we first analyze the Minflux data with a Python code that I wrote, and then feed that data into a legacy MATLAB code that helps track the Minflux, that helps generate tracks with tracking files. So what we see here is what a typical Minflux dataset would look like in the CSV format. So what I did here was I basically converted a .mat file into a CSV just to illustrate what the entire data array looks like. It's simply put a massive array of information. So you've got 26 to 30 columns of tracking information. Not all of it is useful for tracking or imaging. What you have to do is look at what each of the look at what the data structure is, figure out what are the most useful columns. In my case, it's just the iterative localization data, specifically the fourth column, so the fourth iteration, because that's the most accurate iteration, and also the corresponding photon counts and the timestamps for the localizations. So I extract that information and I generate a bunch of CSV files, which I then feed it into the MATLAB tracking algorithm. Then I'm able to generate individual tracks for a kinesin. So here you could see kinesin stepping forward. You see these little steps here. And then these are backward steps, then forward steps again, and so on and so forth. This was in the presence of 10 millimolar ATP. I'll get down to why this ATP concentration is significant. But so this is a representative track. And the tracking algorithm that we have is able to process multiple tracks like this. Sometimes, 10, 20 tracks. Sometimes it could be 100 tracks. Depends on the size of the data set. And then we're able to generate a histogram of step size distributions that'll in turn help us observe the average stepping length. So in this case, we're able to obtain a distribution. We care mostly about the forward steps. So here we're able to obtain 16 nanometer steps at 10 millimolar ATP. Previous kinesin stepping studies using Fiona setups, in those studies, we had to go down to lower concentrations. So in the nanomolar domain, not millimolar, nanomolar domain to observe these 16 nanometer steps. With Minflux, so far we've been able to observe 16 nanometer steps at the millimolar ATP concentration. So when kinesin walks really fast, it's still able to decipher the stepping information very accurately. So that's a promising start, but our objective is to eventually observe 8 nanometer steps, so to try and obtain a second step size distribution here, and that's something we're working on right now. I'd like to thank everyone for listening. I'd like to thank Glenn and Kingsley for their assistance with these experiments, and I'd like to thank my lab members for their support. Thank you. Any questions?
So if there's no questions, I guess we'll move on.
Yes. Hi everyone, good morning. I'm Yuhan and this is Gopika. We're from Grubler Group. Our group are specifically interested in studying protein behavior in live human cells. And today we're going to share our experience and the latest updates on how Minflux is helping us to solve our problem at a sub 5 nanometer resolution. And with Minflux, we're actually able to tackling the puzzle of protein reaction diffusion in living cells. Now let's get started. So one human cell contains more than 20,000 proteins, and those proteins perform majority of their function in the cell. However, the human cell is not as simple as a test tube. It contains ions, water molecules, DNA, RNA, and a bunch of proteins. So those molecules are forming a matrix of interaction network. It becomes extremely important when we study protein behavior, not just from a test tube, but we should always put them into their native environment for study. So our group has studied protein behavior in their native environment, which is human cell, from different perspectives. We've done protein folding study, protein RNA interaction, and protein-protein assembly. All those studies are in the live cell, and it's on the ensemble level statistically. So it would be really cool if we can look into the same questions from a single molecule level, and Minflux makes that come true. So think about this schematic system. So we have a molecule, our protein here, and another molecule that the protein would like to bind to. And when they are in the cell, there will be diffusion as well as interaction between them. So once they bind to each other, when they unbind to each other, they diffuse very fast. And as long as they bind to each other, they start to diffuse slowly. And once they unbind again, they start to diffuse fast again. So this bind-unbind reaction will be an equilibrium, and this diffusion, fast, slow motion is also going to happen repeatedly. So if we are able to look at it at a single molecule level, we're able to use Minflux to track the single trajectory of each individual protein molecules, then we're actually answering this question, is how are protein diffusion and interaction coupled in living cells? They're not two behaviors that are independent of each other. The diffusion is going to perturb the interaction network, and the interaction is also going to affect the protein's tumbling and diffusion. So by looking at single trajectory, we're able to solve the problem of diffusion and interaction at the same time. Now let's move on to our model system. So our model system is a protein RNA complex. Of course, we also have another model system with the protein-protein complex. So our protein is a spliceosome protein called Yuhan, and the RNA is its binding partner, SL2. This is a really common model system for protein RNA interaction, and extensive studies has been done in vitro and in vivo for their dynamics and reactions. So if we want to answer the question of how are they diffusing and interact to each other in the living cell, then we break this big question into three major tasks and solve the tasks one by one. So the first thing we want to answer is, what is the best dye for us to label the protein and RNA and track them with Minflux? The second question is, can we really track the protein on the single molecule level in the living cell? And the third is going to our model system that is actually track the protein RNA complex and track the protein complex in the living cell. Now let's look at those three tasks one after the other. So first, what is the best dye for labeling and Minflux tracking? We have a list of candidates, and we try them one after the other. So what we do is we have our candidate dye, and we pre-mixed the dye with our buffer, and then we micro-inject them into each individual human cells. And then we bring the live cells to the Minflux and do a Minflux tracking when the cell is still alive. So let's look at some example images from our different dyes. So this is a cell image that we can see on the left channel is the green channel, because we also co-micro-inject Alexia 488. That also answers the question, can we have Alexia 488, like the green dye as well? And the answer is yes. So we also co-injected the Alexia, so we have this green channel. We're able to see the cell from the eyepiece, and it will have a clear boundary and a nuclear boundary here. And on the right-hand side is our red channel, which contains the protein that we are labeling with a red dye. That's something we're going to track on the Minflux. So it also, we can also see like the protein is located within the cell and is about to uniformly distributed. Now let's do the tracking. Well, unfortunately, we're only able to get really small and short traces. That is because the dye that we pick here is auto 647. Auto is known for it's very sticky in the human living cells. So it sticks and was not moving that much. So this is actually a bad dye example, auto. So let's look at the next one. Next one, we are also seeing a healthy cell on the left and has a really clear nuclear boundary. And on the right-hand side, we have our protein streptavidin that is labeled with the red dye and is about to uniformly distributed. And the tracking trace is also small and short. Okay, so it's another bad example, auto 655. Now the third one. So third one, we're seeing three cells on the green channel. However, on the red channel, we're not seeing cells. It's about the same brightness as the background. And of course, we're not getting good tracking on this dye. That's because Alexa 647, when it's on tracking in the live cell, it's not very photostable in the live human cells. So that's also another bad example. Now we really know what we're looking for. We're looking for a dye that is photostable and it can label our sample that we are interested, but not stick to other molecules in the human cell. That's the dye we're looking for. So here is another candidate, CF633. We're seeing a beautiful cell shape on the left. It has a boundary on the edge and also has a nuclear area. And on the right-hand side, we're seeing a streptavidin labeled with red dye. And it's located most on the cytoplasm, so we have a darker area in the middle at the nuclear. And we cropped this area on the edge, so we have half of the area within the cell and half area outside of the cell, and we perform the Minflux tracking on this. And whoa, we got really good tracking traces. The traces are really long, and also the traces are all located within the cell, which means we're not tracking random stuff outside of the cell. It's all the dyes labeled to the protein in the cell. So this is a good dye, the CF633. So remember that. Let's look at one more. So here again, it's the edge of the cell, because now we know we really want to look at the edge for the test. So we are able to tell whether it's within the cell or without. And we're using LD655. It's a free dye. It's not hooked up to any of the protein. And the result comes out to be very good. So we can get really long trajectories, and all of them are within the boundary of the cell. We're not seeing a lot of things here that's outside of the cell. So what is this? This is LD655. So for now, we have two really good candidates, CF63 and LD655. Good enough for our live cell tracking. Let's move on to next step, protein diffusion. Let's take a look at some quantitative data. So again, streptavidin labeled with CF633. On the left is the green channel just to see the cell shape. On the right is the streptavidin located in the cell. And we cropped this area, which is half within the cell, half without. And we're getting those traces within the cell. And we can replot the trace on MATLAB by coloring different trajectory with different colors. So like each trajectory can be stand out. And actually, we're looking at here on 2D, but actually this tracking is 3D. So we can rotate this curve to make it a 3D. And if we zoom in for each individual trajectory, we're able to see actually the trajectory starts from here and the proteins diffuse to here. The entire tracking is taking about 80 milliseconds. And this is another longer trajectory that is taking about 600 milliseconds. The protein is moved from here and go all the way random diffusion and to here. Okay, so if we take this individual trajectory and plot its mean square displacement over time and fit this curve with a linear line, then we're getting the dissociation, the diffusion coefficient of this single molecule, single protein. So we can do the same thing with all the trajectory we have in the view and plot this diffusion coefficient distribution. We can do the same thing in living cell as well as in vitro and compare them. We can see in the living cell, in the human cell, it has a wider distribution of diffusion coefficient than in vitro. That is because the human cell is so diverse, different protein located in different places will give you different diffusion coefficient. And of course, we can pick proteins that are diffuse fast, diffuse slow, diffuse middle, diffuse slow from a different distribution and plot their mean square displacement over time. And from this curve, we're able to tell whether the protein is diffusing is a super diffusion or subdiffusion or a regular linear diffusion. Okay, so now we solve the problem of dye selection, protein diffusion. We're actually going to move on our protein RNA model. We have our protein RNA model system, and we're labeling the RNA with the dye that we selected. Well, we can also label the protein. We can do both sides. But boom, this is where the 3D mirror had a problem. So since today, I know there are some Minflux company people staying here online. So my take-home message for you guys is we've solved the problem of we've collected amazing data in the last 3 months. That is awesome for using Minflux, and we're so close to publish our paper. Our publication are just waiting for you guys to fix the 3D mirror or replace it. So work on that. Thank you. Okay, so my part is like this, and I will have Gopika to present another project that is from single protein diffusion to protein-protein complex diffusion. And the model system she's going to present is a virus that everyone cares. So, Gopika.
Okay. Hi, everyone. So yeah, I'll take it up from here. So in addition to the dye selection and the protein RNA complex that we were looking for, we had another system that we were already studying in the lab, which was a even larger protein complex, which is driven by weak interactions between 120 homodimers of a viral capsid core protein. So here you see the hepatitis B viral capsid, which is formed from these 120 dimers, and it's 35 nanometers in diameter. And why are we interested in studying the viral capsid? So here's a very broad summary, a schematic showing the summary of the life cycle of this virus inside a liver cell. So as we all know, the hepatitis B virus attacks the liver cells, and it causes hepatitis B. Which is, in spite of having vaccines, it kills close to 1 million people around the world per year. So the answers we're trying to, the questions we're trying to address here are certain steps in these life cycle, which is very crucial but still debated upon, because the mechanism is not exactly clear. For example, one of the first questions we're addressing here, especially, is how the virus, once it gets into the cytoplasm, how is it transported to the nucleus? Because that's a very crucial step, because it needs to get to the nucleus to release the genome, and where it hijacks the nuclear repair mechanism and for further multiplication and infection. So that's one thing that we're interested in looking at. So we had this viral capsid, labeled with M-cherry fluorescent protein. So the capsid is decorated with 120 M-cherry fluorescent proteins. So this is a system that we use for our live cell imaging with temperature perturbation. So we tried using the same system for Minflux, since we have the 561 nanometer laser, which can be used for tracking M-cherry. And so here you see the image of live liver cells expressing the capsid labeled with M-cherry. So unfortunately, we don't have the pretty-looking cells we had before, because we used U2S cells, which have better morphology, like aesthetically pleasing morphology in the previous slide. So these are liver cells, and they're pretty crowded, but you can still make out the difference. So the darker regions that you see on the right side, those are the nuclear regions, and the rest is the cytoplasm. And the cells are pretty crowded, so it's all cells covering the dish. So on the left side, we have the transmission light, which is a bright field, and we don't have any green dye here, because here we are expressing the protein in the cell and not micro-injecting. So we don't have an additional dye that we can use to detect the cell. So we detected them using bright field. so that we can see the cell boundary and see the nucleus, and select the desired region for tracking. So here we're tracking actually a region with a little bit of cytoplasm and also the nucleus to actually see how if we can capture the diffusion of the capsid in the cytoplasm to the nucleus. And we did get some traces, luckily, and here is a zoomed-in image of one of the representative trajectories, which was about 1.5 seconds long. And as in case of the Streptavidin tetramer, we get a wide distribution of diffusion coefficients because, again, the subcellular environment is diverse, and the capsid would encounter different kinds of subcellular environments while it's diffusing. And we can also get the MSD and the diffusion for the diffusion coefficients of each of the traces individually. And yeah, so again, we are trying to optimize the system here. We'll try again with, instead of M-Cherry, we can try with the dye, which one of the dyes we had selected, which is LD655 or CF633, and also optimize the expression in the cell. So this was like the first time we imaged the capsid in the liver cell. So we realized that in a live liver cell, actually. So we realized that there are options to decrease the concentration and get to the optimum level we can see, where we can see individual capsids, and with the dye it should give us better results. And with that, we would like to thank Martin, our advisor, and also Zan for their timely and insightful suggestions and help with both of the projects. And special thanks to Glenn and Kingsley for helping us all the way from training us to use the instrument to everyday troubleshooting, which was the major part of our work till now. And again, moreover, we would like to thank the Selvin group, Kim group, and Golding group for kindly providing us with the dye samples for our initial testing. And we also thank the funding agencies. Thank you all.
The take-home message for online people is Minflux is an amazing system to work with. Bring your sample and go test it here. Okay.
So if there's not more questions, we'll move on to Dr. Reza.
Hi all, thank you so much for coming. I'm Reza, a postdoc researcher in Dr. Chow's lab, College of Veterinary Medicine, Department of Comparative Biosciences. Today, I divided my talk to 4 parts. At first, I talk about the background of my research that is related to the meiosis I and the different stages of the meiosis I, and then prophase I especially, that I work on it. And then briefly, I talk about my methodology that I use for Minflux and the results that I achieved till now and my future plan. Here in this figure, we can see the different stage of the prophase I that it divided to 4 stages, leptonema, zygonema, pachynyma, and diplonema. Here we can see that the 2 sister chromatids for the paternal and maternal, that in the leptonema stage, we can see that they are separate from each other, but with the cohesion core proteins, that is STAG, REC8, and RAD21L, the 2 sister chromatids, they connected to each other, and especially I focus my today's talk on REC8 because we have the REC8 mutant in our lab. And then during the zygonema, we can see the starting of the synaptonemal complex, and then in pachynyma we have the fully synapsed region for the chromosome, that it's the homologous chromosome connected to each other, and also crossover happens here. And in diplonyma, we can see that the synapse region separated from each other, but in the crossover region we have the synapse, that this synapse is for transverse filament, that is SYCP1, the protein that I label, and also this red part that we can see, it's the axial element of the synaptonemal complex, that the protein that I label is SYCP3. Let's see some figures for the immunofluorescence for the prophase 1 stage in mouse spermatocyte that I use it. Here we can see the late zygonema, that this, the labeling that also I use, it's the SYCP3, SYCP1, that this figure we can see. We can see the green part, it's SYCP3, that it's not synapsed yet, that they are two sister chromatid, but this region we can see the synapsed region for SYCP3 and SYCP1. Why we see the yellow? Because we have the overlap, and the resolution of this microscopy, it's not enough for separate these two from each other. In pachynyma, we have the full synapse, and we can see all chromosome synapse together. Here in diplonyma, we have the separation, and just we can see some parts that the red part, that is SYCP1, it's the region for the crossover, that is still they are synapsed. And finally, in metaphase, we can see that, completely the synapse region disappeared. Here is the model of synaptonemal complex structure. We have the two homologous chromosome that contains two sister chromatid here. The oval blue shape are the REC8. We can see that it's the blue shape, and also the green part, that they are two layers for each sister chromatid here and here. They are the SYCP3 that I label, and also it's the lateral element. And in central region, we can see the SYCP1, that also this one, they mentioned that, it's the two layer for the mammalian. Here we can see more details about the lateral and central elements. We can see the two sister chromatids for the maternal chromosome and paternal chromosome, and we have the crossover region here. And this one is the electron microscopy that shows the central element and lateral element. Here we can see the distance between the two axial element, lateral element that we have, and we label with the SYCP3. And in the center, we have the transverse filament that is the SYCP1 that I label. And the SYCP1 antibody that I label, it's the C-terminal one, that it's close to the SYCP3. For synaptonemal complex, we have different images that they use with different scope. Here we can see the conventional wide field fluorescence, that the resolution is 200. We can see that the synaptonemal complex, it's not clear here because of overlapping of the axial and the central element together. Here they use the SIM microscopy, that is a structured illumination microscopy. We can see this one is the stage is different. It's the diplonyma, that it's the we can see the crossover as a synaptonemal complex, that in between of the two SYCP3 that shows by green, we can see the SYCP1, that is the crossover region. It's clear completely because the resolution is 100, and as I mentioned in my previous slide, the distance between two SYCP3 is 100. Here we can see the D storm with 25 nanometer, that it's more clear, the two parallel SYCP2 that they used for labeling for the axial element, and in between we have the SYCP1 C terminal. Here, on the basis of this model, 3D model, I'm going to do the research on the basis of this model because this model is for Drosophila and I'm working on the mice. Here in this model, let me just stop it in one point. Yeah, here we go. The green part, it's the SYCP3, the axial elements, and in between the blue one, we have the central element as SYCP1, and these are the other proteins that they have the role in the in making the synaptonemal complex. Then in my project, I'm going to show that we have the 2 layer of the SYCP3 for each sister chromatid, and also for the SYCP1 we have 2 layer of the SYCP1. Because of that, I use the REC8 mutant mice that they cannot pair together because they don't have the REC8, and just we have this part for the REC8. Then in REC8 mutant mice, we can see just 1 layer, and also for SYCP2, we can see 2 axial elements. But for the wild type, we can see 2 and 2 layers for also the central element. Briefly, I explained about the slide preparation for chromosome spread. At first, I euthanize the mice and then dissect the mice and collect the testes. Then I mince the testes on the depression slide and transfer to the 1 milliliter of the PBS and mix well and collect the 500 of suspension to the new tube and centrifuge, remove the supernatant and resuspend with the 0.1 molar sucrose. This is the final suspension that we have, the cell suspension. We can use it for the spread on the cover slip 18 by 18 and fix with paraformaldehyde. After this stage, we have the labeling stage for Minflux that it takes around 3 days because I use the overnight labeling for the primary and secondary antibody. And after doing the labeling, during the 3rd year, we wash and then prepare the slides as Kingsley explained for the Minflux. Let's see some images I could get with the Minflux. As I mentioned, I labeled with SYCP1 and 3 imaging with confocal. This one shows the confocal that we can get with the Minflux. Here, it's the image of packeting stage of the chromosome for the wild type. And we select the best one that we think it's good for imaging. And then here we can see the Minflux imaging that we can see 2 parallel SYCP3 with the green. And in between, we can see the SYCP1. For this image, I measured the different part of the 2 sister chromatid, here, for example, here, for different, because I have some different image for the wild type, and I select 3 images, and I measured the axial element widths, and then compare with the REC8 mutant that just they have the sister chromatid. Here we can see the image for the REC8. Also for REC8, I measured the widths of the axial element in different parts and different slides. And then I compare these, the widths of the SYCP3, that is the axial element, and we can clearly see that the widths of the REC8, because they have sister chromatid instead of the homologous chromosome, we can see the thinner widths for the SYCP3, and this one is the significant. Here is the model for the REC8 that we think that it will be happen for the imaging, but because we don't have any imaging for the REC8, just SYCP1 for the central element. For the wild type, we can see the 2-layer of the SYCP1 as a central element, but we think that, yeah, if we get the image for SYCP1, we will have this result for SYCP as a central element. Just 1 layer, we can see that connect the 2 sister chromatids together. In next several slides, I will show you the some images for different proteins that they have the roles in the synaptonemal complex. It's RPA in combination with the SYCP3 as a axial element, and we can see the RPA replication protein A that we can see this that. Just I get this image 1 time for RPA, and it needs to get more image to get the more details of this protein. Here also we have the SYCP3 crossover region. This image is interesting. Also I did for the REC8 mutant, this one, that we can see that in the crossover region we have the separation between the sister chromatid. Here we don't have any data acquisition. It shows that maybe during the crossover we have the separation, the break between the sister chromatid, and again they connect together for exchanging the chromosome sister chromatid together. Here also we can see the same pattern for the sister chromatid in the REC8 mutant mice, that we have the separation and maybe it's a break between the sister chromatid. I'm going to do some maybe 3D images, because we can see in more details that if we have any exchange of the chromosome between the sister chromatid here. For my future plan, I have planned to do this, the staining SYCP1 and double staining of SYCP1 and 3 between the wild type and the REC8 and compare these two. Also, as the other presenter they mentioned, using the nanobody, maybe it can be helpful for getting the better image for the SYCP1 and 3. And also, I'm going to check the other synaptonemal complex, because in our lab we have different mutant mice that they are knocked out for different proteins that they have the role in the synaptonemal complex. Then I can compare those mutant mice with the wild type. And I acknowledge the IGB Core Facility Center, especially Glenn and Kingsley, for their training and help for slide preparation and learning the Minflux, and also Dr. Chow's lab and all members, especially honorary member of our lab. Thank you so much. Thank you for your attention.
Some other questions?
Yes.
Yeah, so very good job. So
Thank you.
I didn't get a sense of the significance of looking at the different structures and what is the ultimate goal and what are we trying to show?
We want to show that the REC8 has the sister chromatid, and the wild type, it's the homologous chromosome that they are pairing together, but for REC8, just the 2 sister chromatid, they pair together.
Okay, so in the overall scheme, how would that information be helpful to us in understanding the chromosome division? And does it have any implication on birth defects or anything?
No, because in this result, actually, we know that REC8, it's the sister chromatid that they pair together, but we want to use this one because nobody reports that this model for the REC8 as a SYCP1. Then for SYCP1, we have planned to label just the single labeling of SYCP1 for the wild type and the REC8, and we can measure the widths of the SYCP1 and then compare with the wild type. If we have the SYCP1 for the connecting, for making the synaptonemal complex in the REC8 also.
Okay, so I guess my point was, most of the time when we are interested in studying the chromosome architecture is because any defects in how the material is exchanged can lead to birth defects, right?
Mm-hmm.
And so my question then is, these specific measurements, how does it impact the overall scheme of understanding of the chromosome architecture and possible defects that can arise?
Yeah, actually for this one, because it's REC8 and REC8 mutant, of course we don't have the core cohesion proteins, and it's not possible that we have the two sister chromatic pairing together, and we have the synaptonemal complex for the REC8 one. Because of that, it affects to the REC8 mutant and cause the infertility in the mice. Is there any other question?
Yeah, so just to add to what Reza said, this is an area that I study for my PhD. So I just want to add that until Minflux, the process that we've had an idea of the structure is by TDM, but at Minflux we can now look at the actual two systems. So there's homologs and two systems, and the two systems are also separated. And we never had measurements of those. Minflux is able to resolve that, and clearly we can see two sisters as well as the homologs when they come together and how they exchange genetic material.
Sure.
That is very significant.
Sure. Thank you. Thank you so much.
So we're now scheduled for a coffee break. And then in a few minutes, we're going to start a demo that Kingsley's going to run. So there's two options to watch the demo. One is to stay here. We're going to have the whole system running on Zoom, so you can stay here and watch it on Zoom, or you can go down and stand in the room with the Minflux. If you're going to stay here, I would encourage you to go look at the Minflux at least initially. The reason we're doing this is because the room might hold between 5 and 10 people, depending on how close you want to stand to other people. So if you don't want to stand very close, I would suggest that. Anyway, so that's what we're going to do next. If there's questions about that, if you aren't familiar with the core, grab me or Kingsley or Austin or Dewey, and we can help you find your way down there. I would also like to thank all the research speakers again, because I want to point out that the images we just saw would not have been possible to come out of the core without their hard work. And also these are a barrier in the Minflux system, because these are a step up of resolution that we've seen before. Yeah, thank you. That's great. Thanks. Just jump through a bunch of them. So for people on Zoom, we're on a coffee break now, and in a few minutes we'll come back with Kingsley running the Minflux on Zoom also, and you'll be able to see that. So just hold on for 5-10 minutes. There you go. Thank you.
Hello. Yeah. Nice to meet you. So, Yang and Ray, and he's actually checking out. We're three super.
Right, I saw that. So, yeah, so welcome down. You want to go look at it?
Yeah, some people from Abberior. Okay. Yeah. No, that's fine. I just. Was it the sharks?
Okay. Try to click share screen. Yeah. Okay.
Oh, Tommy, there's going to be audio, right? No, I don't get audio from there. Maybe I need to turn something on here? Oh, that makes sense. There's the problem right there. Kingsley, can you say something?
Oh, it's time already.
Okay, no, I just
I'm
wanted,
just trying to
I just wanted to check.
reinitialize the software. Just give me a second.
That's fine. I just wanted to check. It works.
Okay.
We were just checking the audio.
Okay, so
Yeah, I'm gonna lower it a little bit.
I think we are ready to go. So
Is that okay?
typically you start the software by clicking on the Minflux tab, and then it opens up. One key thing that you want to look at for is the daemon. So the daemon here is basically a software that monitors and makes sure that everything is working fine. And so once you don't get any error message in the daemon window, then it means you are good to go. Oftentimes, you may notice an error message. You may want to close and relaunch it. So you can close it here and relaunch the daemon right here. And it will go through again and check and make sure that everything is fine. There's been a few occasions where you may have to actually close and restart the entire software. And to some extent, sometimes there's a loss of communication between the Olympus system and the Minflux. And so when there's a loss of communication between the Olympus system and the Minflux, you may have to do a hard restart. But that happens occasionally. But most of the time, everything works fine. So I've just loaded the software. And before I mount a sample, I just quickly want to explain the few windows that we're seeing. So this is a Minflux acquisition window where we go to select whether we want to do tracking or imaging. And so we have different sequences. And as Glenn mentioned, some of these sequences can be custom modified for your research. Most of the time, the standard ones work fine. You have the stabilization window where we can look at the beats and stabilize them. We have the channel windows where you can set up all your channels to do confocal as well as to do Minflux. And here, whatever settings that you have is basically summarized in the measurement window. You have the scan rate window where you can set to image in a confocal mode. So in a confocal mode, you can image about 80 to 80 micron in XY. And so you can increase it to up to confocal maximum of 80 by 80. If you're doing Minflux, you crop a window. And so oftentimes you want to do within 10 by 15 micron. And here you can adjust your pixel size as and when needed. And so the size of the pixel will depend how fast you are scanning. And for people that may be interested in doing a remote control of the instrument, so here are the XYZ controls. So here you can use your mouse and move the stage in X, Y, and Z to focus and to set up to image. Down here, we have the Minflux data panel. And so in the Minflux data panel, you can render your image at a specific pixel size. These are the different measurements that will be recording. So Glenn mentioned some of them, the center frequency ratio, the detector channel ratio, if you're interested in two-color imaging, and all the other information, including iteration for people that are interested in tracking and other stuff. So all these are all different measurements that are recorded. And so if you are interested, you click here and it can explain to you a little bit more of what each one it does. And this panel is the line step panel. So if you're doing a confocal imaging of two channels, then you want to have two lines. For Minflux, essentially one line is okay. And so we can have 2 or 3 lines, and then we can also select how fast each line is being scanned. Down here is the control for the IS-83, that is the Olympus part. Like I said, you can control this manually here as well as through the software. And the display tab allows you to display your image in XY, in XZ, and YZ when you have some data. All right, so what I'm going to do now is I'm going to mount a sample. So Glenn is going to try and show you with the camera. Yes, I'm sharing.
Ready.
Okay. So this is us. Yeah.
That's nice.
All right. So this is the Olympus part, and so this part is like any other microscope, and of course this is a Minflux. It's a 10 key, so everything is controlled. When you turn on the software, you don't have to turn on the individual parts. It's just one click and everything is on. So you want to add your oil to your slide as you do for any other imaging. And so I invert, because it's an inverter system. And so I invert my slide onto, and then I clip it with these clips to keep it a bit more stable. I adjust my Z position to make sure at least the oil, I'm at the cover slip. And then you can bring this down. So if you want to have an idea, you can raise it up and have an idea where your slide or the objective is relative to your sample, and then you can close it back if you don't want any light going through your sample. Now, in the bright field mode, you can look through the eyepiece, and then obviously focus on your sample. Here you can do bright field, you can do normal fluorescence. So there is this control where you can switch between bright field and fluorescent mode, and then look at your sample, whether using the GFP or 488 or 561 before you actually go into the software. Now, all that is not necessary, because you can also control everything through the software. And so all that we need to do is just mount the sample, and then you can do the rest through the software. So we have several templates that we can use. We can actually create a new template from the scratch, but for the sake of time, I'm going to skip that part. So I'm just going to load Minflux templates. So things are a little bit slow because we have live Zoom imaging as well as a big screen connected. So things are going to be a little bit slow. So please bear with us.
Yeah, that's right.
It's taking a little longer than normal to load. Once everything pops up, we'll be ready to go. Typically, it doesn't take this long. Let me minimize this back.
That region is not good actually, because he's talking about.
It looks like it's frozen or something.
Okay.
All right, so somehow it's okay. Let me see. I need to go back to 1 and redo this again. So the software is frozen on me. I need to relaunch and hopefully get it resolved. All right, so this is the problem that I mentioned earlier on. Sometimes if you switch in between bright field and looking through the eyepiece, there is a filter here that you need to switch to 1. And so if you don't switch to the appropriate position, the software may lock and you get some of these errors. And it is very annoying, but oftentimes you can reinitialize and get going. So I apologize for this, but it is a real situation that happens from time to time. By the way, so whilst we wait, I'm just going to tell you a little bit about the sample that I've just mounted. So it is a mouse chromosome sample that Peza was talking about earlier. And so we have 2 color labeling. We have SCP3, that is the label for the outer core of the chromosome architecture, labeled with 647, and then the inner core is labeled with a 680. So it looks like we are ready to go now. Let me try again. Okay. All right. So currently, I have my Minflux window set up, but I'm first going to do a confocal where I'm going to look at my channel. And so you can set up any number of channels of interest. Let's say I'm interested in 2 channels. The first channel being a 640, and then maybe a second channel, say, let's say 561, okay? And so you can set up to image based on whatever 2 labels. I've increased my citation for the 561 to 2%. This one is at 1%. Maybe let me go to 2%. All right, so in this window, then you can go live. And it takes a second, you hear a click. So switching from the system, I have another error. This is not good. Okay. So let's set the switch. Everything was working perfectly this morning. Okay. Let's try again. I really apologize. I'm not so sure why it's giving us, because we tested this with a Zoom connection yesterday and it wasn't this slow. Today is just so slow. So I really apologize for this. It's completely frozen on me. Okay. I may have to do a hard restart. I apologize for this. Software completely frozen. It's not liking all the connections for whatever reason. Okay. Yeah. I'm going to do a hard restart. Yes. So that should solve it once and for all. So, like I said, this happens once in a while. It's just unfortunate that it's happened today, but it's occasionally we encounter this. So I'm giving it about a minute or so, and then I'll turn everything back on. First, turning on the Olympus part. All right, so we give it about a minute for it to reboot, and then we turn back on the inspector software to turn on the Minflux part of the instrument. Someone has a question online. It says, with the use of glass buffer, how long are you able to maintain live cells for imaging? So typically the buffer will work for about 6 hours to overnight, by which time you would have probably degraded the stability of the enzyme. So typically 6 to 8 hours, but there have been occasions where we've been able to image even overnight. Okay, so Demon says we're good. So let's try again. All right, so to focus, we press F9, F10. And so I'm adjusting in Z to get close to the cover slip. So once you restart everything, it goes back to 0. Our focus point is around 6,000 in Z. So I'm quickly going up. Those of you online will not see that. But once I get to close to 6,000, and then also you can see the beats. So we added beats to the slides. And so as we get closer to the cover slip, you see the beats. And so now I can see I'm closer to the plane of interest. I want to increase my scan area to 80 so that I can scan a larger area. Now I need to move around and find cells. These cells are kind of spread out a lot, and to avoid high background, we often don't put too many cells on the cover slip. So it will take me a few seconds to find a good cell. I believe this is probably a good cell. All right, So then I can choose a region of interest, assuming I want to focus on this cell right here, in this chromosome. I want to image this chromosome. So then I can clone it. So I clone that view. And then I want to decrease my pixel size so I can get a better view of that. So I'm going to do 40. And so then this allows me to do fine focusing. And so you can do fine focusing, either a course or fine focusing through the software. So once you have the area of interest well in focus, you can pause it, and then while this is going on, you can also do your stabilization. So we see the beats. We turn on referencing. So once we turn on referencing, the camera is looking for the beats, and once it's able to localize the beats, it puts a mask, and then it tells you to lock it. So this is the X, Y, and then the Z. And so telling us that we've stabilized it within 0.4 nanometers and about 0.6 nanometers in Z, which is perfect. And this stabilization can last oftentimes hours to even a whole day sometimes. But it's important that there is the room, there's not too much movement in and out, or the doors nearby are not being banged. And so you want to keep an eye on that. All right, so now we have a region of interest. There are two ways that you can image this. Once again, I can make another copy of this same window. So I can have a confocal image, and then I can have one that I'm going to set up my Minflux on. And so within this region, my focus is this particular chromosome. And so I can draw a box, okay, an ROI, we call it, region of interest around the entire chromosome or a particular region of interest. And then once we have a region of interest, I want to once again clone it. So I can relate the exact area of my confocal image to my Minflux image. Okay, so now I have the exact area of interest for my confocal and then Minflux. Now what I do next is that I select the entire box or entire region, and then I set it as Minflux ROI. And so now you see that the mouse has changed to green. So anything that I'm doing here is no more from confocal, but I'm in Minflux mode. And so I go here and select the sequence with that I want to use. So we have 2D sequence and 3D sequence. So I'm going to use this sequence IF, which is our 2D sequence. People that are interested in 3D, when we have our mirror fix, we'll be able to select the 3D sequence, which is this guy up here. All right, so now essentially we are ready to go. And so then you press on the Minflux record button here. And then right away you see blinking. So earlier on in my talk, I indicated that sometimes it might be good to bleach your sample a little bit before you start imaging, so that you get single molecules. I didn't do that because we are already behind time, and so I'm kind of rushing through this a little bit. But currently I'm citing with about 1.5. Typically for 2D, we do about 2%. And so what you see here is some numbers. The first number that we see here that is of interest is the valid localizations. Already we are in about 2,000. It doesn't necessarily mean that we are able to localize all this. We have the actual traces being only about 15. So that means that we can localize a molecule, but by the time you go through the iterations, another molecule might be excited nearby, and so it will consider that as not a valid trace. It will ignore it, or even the molecule may bleach before it goes through the iteration. And so we have a much smaller number of valid traces compared to the actual localizations. Even for the traces, it is not all this number of traces that actually lead ultimately to a valid image, because some of them can be the same molecule that after localizing, it goes off, and then later on it can be activated back on. And so some of those traces can be the same molecule. And so all of them are not necessarily distinct localization. Then you have the catch ratio. Basically, it goes from none to sometimes 10%, 50%. It changes. And so it gives you an idea whether it is able to find the molecules to localize. And you have the event time, and then the effective frequency. So these are the frequencies of the localization. This is one way that you can plot your data. Now, after maybe 5 or 10 minutes, you see that you may not be getting enough localizations. And so then you turn on your 405 laser to bring the molecules back from the dark state into fluorescent state. And so now we can start increasing 405 laser. And so here it depends on your sample. You may go from 1% to 2% every 5 or 10 minutes, or it depends on what you're doing. Sometimes you may have to go up to about 20% by the time you finish imaging. And so currently I'm at 2%. And so I'm getting nice blinking. These numbers, oftentimes you want to have some range between about 40 and 100. So as long as you're getting localizations, you are good. So we should begin to see the localization as well as the detector channel ratio. Now, because I have two color, later on I will be talking about how we can separate the two colors. So currently I'm imaging and you may not get a clear picture, but with time, we're going to see a trace that focuses on just the region. Now, this area that I chose has a lot of background. And so it gives you an idea how the effect of the background can also affect your image. So in your labeling, make sure that you minimize a lot of the background, because all those backgrounds can be picked up. So now another issue that you should keep in mind is also your MEA concentration. So oftentimes for your sample, you may have to titrate the MEA concentration. And so you may start with about 4 micromole of your MEA, maybe go to 8, go to 12. And so oftentimes you may have to titrate it to get the best imaging. So whilst all this is going, we can actually start looking at our data if we have enough localization. It's taking a little while to get enough localization to mark the track of interest. But we have this window right here that also tells us all the data that we can collect. And so one important software that I did mention in my talk is ParaView. And ParaView allows us to visualize our data even whilst we are collecting. Obviously, we can see the localizations here, but you can also visualize it in ParaView. And so once you click on Launch ParaView, it's going to launch the ParaView, and then you see whatever localizations, or even as you collect the image, you can get some localizations here. And so it can give you an idea whatever you're trying to image. Even you don't have to wait 1 hour, 2 hours to get a fair idea of whether you're able to get good localizations or not. We're doing on time. All right, so we may need to give it about maybe 15, 20 minutes for us to get enough localization to get a better picture, but this is still ongoing. Right here, I want to talk briefly about some of the information that we can obtain. So we have the center frequency ratio, detector channel ratio, effective frequency at offset, background frequency, the iteration, the localization, the status, and then the ID tracer. So the ID tracer is basically important for people that are doing tracking. The status is most importantly for checking the system if there's a problem. And so typically the Abberior people will be more interested in that. Localization, this is the individual coordinate. So each time you localize the molecule, the coordinate is recorded. And so that the coordinate can be plotted as an image for you. Also the iteration index. So each time you localize a molecule, you go through 4 iterations. And so each of the iterations, there's an index and you have data in each iteration. So the iteration index records, keeps track of each iteration and the kind of data that you recorded at that. Then you have a background localization. And so here the software uses that to know the background as well as when you have an activated fluorophore. And so when to ignore those background and then focus on an active fluorophore and try to localize it. You have effective frequency at offset. Now this is important. What this simply means is that in your search pattern, let me just increase my 4.5 a little bit as I keep talking. All right, so the effective frequency at offset, basically you have localization in the center of your search pattern as signal from the center of your search pattern as well as the periphery of the coordinates. So we are looking at the ratio between the signal from the center to the ratio of the signal at the periphery. And this is an important determinant that can be used to discriminate a molecule that pops up whilst you are actually localizing another molecule. So oftentimes the molecules can turn on and off randomly. And so if you are trying to localize a molecule going through the iteration, and maybe by the time you get to the 4th iteration, another molecule pops up, then you're going to get a higher signal from your donut because of the new molecule that shows up. And so the ratio is going to change, and so the software is able to do that and then abort that trace because you don't want to have 2 molecules turn on at the same time and it will not know where to which one is localizing. So that is a way to discriminate multiple localization at the same time. Then you have the detector channel ratio that we use to separate 2 color channels. We have the center frequency ratio, which is also important as another determinant in characterizing our image. So I'm going to let this go in the background, but I want to open a typical data which I took yesterday, and then hopefully we can go through that because I want to also show you the tracking. So if we want to wait for this to finish, we might probably take a little longer. So let me pause this and then open data that I took just yesterday from the same data set that we're looking at, and then I can explain a little bit more. So it's taking a little while to load.
standard sizes of acquisitions for like 2 week?
You mean the area of?
No, like the file size.
Oh, it varies. It depends on how long you acquire the data. So oftentimes, I'll just quickly show you one of those, and then we can move on. So this one is about 138 megabytes, and it's data that I acquired, I think, about 2 hours. And so if you are imaging overnight, obviously the size will increase. So it depends on what you're doing. For tracking, it may be even much less. Anyway, so this is a typical image. In fact, this is the same slide that we are currently imaging, an image that I took yesterday when I prepared a sample. And so this is a region that I chose. And so we have the confocal image, we have the detector channel ratio, and then we have all the localizations. So from this image, we can then launch the ParaView. And what we want to do now, and I quickly want to show you how we do the 2-color separation. So if I launch this in ParaView, it should pop up. Hopefully I selected the right file, because let me go back and make sure I selected. So right here, you have to indicate the particular data that you are analyzing. So the data that I want to analyze now is 221, I believe, which is what? Let me see. Yes, 221. So I need to point it to that right data and then launch the ParaView. And so once again, it's going to take a few seconds to load up. Now, I know we have Abberior people online. So as we're going through all this, if you have any questions for us or for Abberior, there are guys online that can answer any question beyond me and Glenn. There will be a question and answer time where you can ask all your questions. So please keep that in mind. Yes. I'm sorry? The cost? Oh, well, that's an interesting question. So the imaging cost, we're charging $60 per an hour. Yeah, so in terms of the imaging cost, that is the price. And typically for imaging, you need about 2 hours to get a decent image. So maybe with about $120, you get a very good image if everything works perfectly. But sometimes you may have to go a little longer, and so that can add to the cost. Obviously, for tracking people, they may spend much less, but we still charge the same rate.
Kingsley, do you have time for one question?
Yes.
So in the chat, this is maybe referring to something previously, but with the use of Glock's buffer, how long are you able to maintain live cells for imaging without introducing hypoxic stress?
Yes, I did answer that earlier
You did.
on. So
Okay.
the gloss buffer can last about 6 to 8 hours, but there have been occasions where we've been able to image even overnight.
So it's taking a little while for the software to load up, and we are waiting for it. Once it load up, I'll go through the process again. But this is the image that I just showed you. This image. This is how we separate it in Inspector, and so this is what I wanted to go through. I'm show you the final product, and maybe if we have time, I can show you the exact process. So you load up your image, and typically it will not look like this. This is now the final separated image, but you will then come here and then add your histogram. And so based on the histogram, you can then look at the detector, the various signal that are collected. And so we see that we have clearly two peaks, one being on detector 1 and then one being on detector 2. And we see that they are separated around between 0.35 and 0.4. So we then set, we create a color map where we are looking at signal from 0.1 to about 0.3 right here in red, and then another one from about 0.4 to all the way to the end in green. And so if you do that, you get a clear separation between the two channels. And so here we have the red channel marking the chromosome that is the outer core, and then the inner core being the SCP1 clearly separated. And so if I'm able to get this up in time, you see that it's still working. Like I said, we have 3 connections, a big screen here, and then also on Zoom. So it's slowing down everything a little bit more. So everything is taking a little while. But I also want to do tracking. So if I realize it's taking too long, basically the point is what I've shown you, and you can clearly separate the two channels and get your 2-color image. And it doesn't matter whether it's 2D or 3D, it's the same process. Another thing that I wanted to show you, if we had time, is that you can also do your image by masking. So these chromosomes, some of them are curved. Some of them may be such that if you are imaging the entire region, you have a lot of noise. And so you can set a mask where you basically tell the software to only look for signal in your mask region. So basically this will be decorated, and so it will only search within this region and not search outside this region. And that also can speed up your imaging, and you can actually get very accurate data from that. So unfortunately, I'm kind of running out of time, so I might not be able to show you that today. Okay, so now it is loaded up, and for some weird reason, it's showing up as if we are doing tracking. So I turn off I turn off that. So this is the data now. And so the first thing that I want to do, let me close. So if we were to do tracking, all this will be tracking information that we can utilize. Now I don't care about that for now. So I go to my localization, and then I add a filter or go to my favorites. So I've put that in histogram as one of my favorites. I go to that, and then I want to switch from the center frequency ratio. So the kind of localization that we're looking at now is the center frequency ratio. It could be the localization. They will all look similar. You might not be able to tell the difference. So if I go to the localizations and apply, the image looks the same. All right. But what I want to do now is to go to the detector channel ratio, and I want to increase my bin count to maybe about 40, and hit apply. Actually, I should be doing that with the histogram. So where is my...
this one, yeah.
Kingsley, do you have time for another question from the chat?
Yes, yes.
For two-color Minflux, it was mentioned the sign of the fluorophore based on their to assign the fluorophore based on their peak signal. In this case, how do you manage the bleed-through effect? How does this affect colocalization results?
Can you repeat the question again, please?
For two-color Minflux, Lexoflora 647 /CF 680, it was mentioned that you assign the fluorophore based on the peak signal. In this case, how do you manage the bleed-through effect, and how does this affect colocalization results?
Yeah, so so that's a good question. Your dyes, you have two dyes, 647 and 680, that are all excited by the same laser. And so, as we explained in the talk, okay, so I don't have that image here. I thought I had that image here. All right, so you have two detectors that are both detecting, and so it is using a ratio of the signal that is going to each of the detectors. And so if the ratio is, let's say, below 60%, it will assign it to one particular detector. If it is higher, it will assign it to one detector. And so if I show you this image, clearly you see a region where there's an overlap. There's an overlap. So the signal in this overlap region, will have to ignore it. And so that is what we do the filtering, whereby we ignore the signal from where there's an overlap, and we are only focusing on signal that is close to the peak of this detector, and then signal that is close to the peak of this detector. So that is how we are able to separate them without any breakthrough. I don't know if that answers the question.
Yeah, I think so. The other thing I'll say, just conceptually, this is a little bit different than standard microscopy, right? Because this is just for one fluorophore, right? So it's there's not going to be, you're just assigning it based on that, right? You're categorizing one fluorophore at a time, so it's not going to be quite the same as a confocal image where you have populations of fluorophores and there's crosstalk and you can't discern those.
Yes, thank you. All right, so here is the image again. This is our image, and this is our two peaks. And so once again, what we are interested in doing is now try to go to our DCR, and clearly, if I am able to move this, you can see that there is this peak. And this peak probably ends around 0.35. This is another peak that probably around 0.4. And so what I would like to do now is to bring up my map. Typically it shows up automatically. Let me see if I can bring it up. Where is this? So I'm not seeing my map. Just a second. Anyway, so this is what I'm trying to, my color map editor. And so typically this pop up, there is a way to bring it up, but so I think it's this window right here. So edit color map. So I want to see the same thing in the window that we are working with. So it's kind of grayed out here. So if there's a way I can turn it back on. I'm not so sure why it's grayed out. Oh. Everything is so slow, so. Yeah. Right, so now it's up here for me now. All right, so we are good. Okay, so there's something weird about the one that opens up. I don't know why. This one. No, that's not what I'm looking for. Typically this one pops up. But you see, I made a mistake of adjusting it wrongly. So I don't have the separation tool. Okay, so I should be able to have this. So what I'm trying to get today is this. Yeah, these two. Okay, so typically it will show up like this, and then we define the region. And so just to show you, you can have different color maps. And so you click and then define the color. And so I want to have within this range being red. So I can assign red color and I click OK. I double click this, I assign the red color, I click OK. And then I want this to end around 0.35. And then next to it, I want to have a black region. So once again, I want to have 0.35 around here. I click on this, I assign that to be black. I do the same thing, I click on this, I assign that to be black. And then between this region, I want to have the net separation. So from 0.45 all the way, I double click on this, I assign that to be green or any other color that you want. Let's say I want that to be cyanine. I can select that and then I can adjust this to be cyanine again. Okay, so in that case, I have my two color separation. So I have red and then the cyanine in the middle. And so that is how we separate our two colors. So I've taken a lot of time on imaging, but there's a lot that we can discuss. So I want to quickly switch to my live cell and show you a little bit about tracking also. And once again, whilst this is going on, if you have any question, you can ask. So I'm going to close all these imaging tabs.